So this is the title of the trial that is proposed. It's a combination phase 1, phase 2 with some efficacy endpoints as secondary outcome measures, and it's titled A Trial of Diaphragm Delivery of Recombinant AAV Acid Alpha-Glucosidase in patients with Pompe's disease. It's sponsored by the NIH and our center at Florida, and myself and Catherine, who was here previously, are the principal investigators. I'm just going to cover the aims of the overall study, the rationale for it, you all know the background quite well, so I won't really dwell on that. Go over some of the preclinical studies that support this, including the biodistribution and toxicology studies that have been completed thus far, and then go over the protocol specifically. And I hope I can do that all in about a half an hour. So first of all, the aims of this study are to assess the safety of this approach. So all early phase studies are really aimed at understanding safety and not having primary endpoints related to efficacy. That's been true of most gene therapy trials, have been phase 1 or phase 2 studies. And particularly in the AAV field, there is only one late phase study that is a pure phase 2 for a neurologic condition. So the safety measures are really to understand the biodistribution and particularly assessments related to immune response to the vector itself and or to the transgene product, which in the patient population we propose a study in, already a good bit is known about their response, at least to the recombinant protein from the standpoint of their humoral immunity, but not a lot has been done. In fact, there's been no studies, systematic studies of the problems associated with T-cell response. And then we're going to look at the dose-finding effect on pulmonary function. And these patients will all be receiving concomitant use of Myozyme. And what we're going to assess as the secondary outcome is the effect of the gene transfer on the diaphragm and the phrenic function. And I'll go through the rationale for why that's being used concomitantly with Myozyme. So we know this really very important study done by Priya to establish the natural history of the disease was really the mark which we compared all the treated patients to. And you can see the severity of the disease when you look at this in the early onset patients. There is a subset though, there's a subset though that are still alive even after a year, and that patient population may be some of those that would have presented later prior to the advent of really assessing patients comprehensively and giving them the opportunity of treatment with Myozyme. And we knew that this really full spectrum of disease, somewhat represented by that natural history curve in early onset patients, was inversely related to the amount of enzyme activity. And patients with the most severe condition have no enzyme at all, and up to the carrier state, which are asymptomatic. And somewhere in between there's a representation of this spectrum in all the patients. But one thing that's become clear in understanding the new natural history on Myozyme is that there's a progressive loss of independent ventilation even in the children. And I think we know the mechanism of that now. So the goals, as you can imagine, is really to get the enzyme into this compartment. That happens normally when protein's made in the cell itself, and a good part of it goes through the mannose-6-phosphate receptor that's on the Golgi and traffics through the vesicles and the transport vesicles to lower the pH and reach the lysosome. That mannose-6-phosphate receptor is recycled within the cell, and that's the most important mechanism by which lysosomal proteins are targeted to this compartment. Some of the protein can be taken up by the cell surface receptor, and that's really the basis for enzyme replacement therapy. A very small amount of that process, we think, has some limited efficiency, and certainly this intracellular route is really the means by which most protein reaches the lysosome. And that may be another reason, besides, as Paul mentioned, the large muscle mass, the fact that this is not a highly efficient process may be responsible for the dose needed in striated muscle as a target. And every striated muscle is, in fact, a target, and we see the effects of GAA deficiency in all tissues. A small amount of the protein can be released from the cell. It's not a very specific process, but there's some evidence now that a bed of tissue which has a correction by the gene can be a source of recombinant protein, and then you're back in somewhat of an enzyme replacement scenario where that protein can then traffic out of the cell to another nearby cell, or no need for it to go to the cell it came from. So in these studies, we were really encouraged by the fact that the response in the patient population and infant population was quite dramatic. And I'll use the kind of high overview of this data to show you what were the keys, takeaways from some of that in terms of what are still the unmet needs of some of the patients. We had a group that was identified before the trial started that were in this age group of 6 to 36 months. That was actually the first trial started, known as 1702. The hope that there would be a more homogeneous population when they were identified a little sooner was the basis for 1602. The primary outcome measure of the 1602 study, though, is ventilator-free survival. In 1702, since patients could be on a ventilator at the start of the study, survival alone was the primary outcome measure. We always have to keep in mind that when we try to merge those two things together, we're mixing two endpoints between the two studies. But we'll really look at ventilation as one of the key outcomes in both studies, because even patients in 1702 who initiated the study without ventilatory support, some became ventilator dependent. And my background as a cardiologist was to be mostly interested in the heart disease, and I've been diverted from that because really I think the favorable response in myocardium is primarily responsible for the improved outcome in the patient population. So if we look at what are the other factors that limit patient survival, it's this need for assisted ventilation in young patients. There was a dose difference in the 1602 group that did not have a big impact on outcome in both the time period studied and subsequent to that. The higher dose was only associated with more infusion-associated reactions. So here's the really good news, is that in the 1 year the patients were studied initially, all of them in 1602 survived. So this is a milestone, really, in really being the first neuromuscular disease, a fatal neuromuscular disease, to have a very specific treatment which was effective. And I think this is the basis for the licensure of the drug in the young patient population. And compared to those controls, where only 1 of 62 would have survived.